Good morning, everybody. This is 2.8. This is cell respiration. I'm going to do my best to write neatly, but please listen and watch multiple times because I do have messy handwriting. So, the SL portion of respiration is really straightforward. The basic definition is going to basically be the controlled, keyword is controlled release of energy, okay, to produce ATP. Now, ATP, this is a review. ATP, we should remember, is adenosine triphosphate. It's frequently referred to as the energy currency of the cell. All things in your body require ATP. When you say this requires energy in the body, we're talking about ATP. So generally, it's drawn like this. This is your adenosine. This is the same molecule. This is ATP. This is A in DNA, right? So a nucleotide, right, is you have your phosphate group, you have your 5-carbon sugar, and you have your nitrogen base, right? So this is adenine, guanine, thymine, cytosine, right? This is your ATCG, right? And when you're making DNA, you have ATP, GTP, CTP, TTP, right? So all these can form triphosphates. The body uses adenosine phosphate. So if I take this nucleotide and I flip it around, we generally draw ATP like this. So we have adenosine, there's your phosphate group, and then this is your high energy bond, phosphate group, high energy bond, phosphate group. And ATP cycles with ADP. So this is adenosine, ADENOSINE, phosphate, high energy bond, phosphate. So ATP is oxidized to ADP and reduced to this is oxidation, oil rig. Oxidation is oil rig. Oxidation is loss. Reduction is gain. So ATP is oxidized into ADP and ADP. So this is ATP, ADP, right? So this is the first section of SL respiration. You just have to know ATP, ADP, okay? You need to know that ATP is the main energy source. Your body also uses GTP, but not very much. Sometimes it will even take ATP, it will oxidize it into ADP, then oxidize it again into AMP, adenosine monophosphate. Basically, this is a nucleotide, right? And then it goes back to ADP and back to ATP. So, but ATP is the main energy source. That's all we need to know, okay? So now, the next concept is you have anaerobic and you have aerobic, okay? Anaerobic, aerobic. ANAEROBIC, AEROBIC, right? This is without oxygen. This is with oxygen, right? This is obviously, aerobic is more common and it's more efficient. Okay. Aerobic has a larger yield, so this makes more ATP than anaerobic. And this also connects back to evolution, right? So anaerobic evolved first. This evolved first, and then aerobic evolved second, and then aerobic took over the earth. Aerobic bacteria is what changed the primordial earth, right? There was no oxygen in the primordial earth, in primordial old, right? Very old earth, right? So as the aerobic bacteria began to populate the earth, they helped change the atmosphere. So they changed the atmosphere from an oxygen absent environment to an O2 rich environment. So you can honestly you have to thank aerobic bacteria for evolving, which literally changed the atmosphere of the earth. This is important. This connects back to evolution. However, it's not part of it's not part of 2.8, but it comes up other places. So anaerobic, all you have to really know is the word equations, right? So aerobic, it's more common, it's more efficient, it's a larger yield, and it comes from glucose, right? Now, anaerobic, there's two types, right? Basically, we have lactic acid or ethanol, right? This is in animals. This is in plants. Okay? This is used in baking, right? So yeast, they take glucose and they produce CO2 and ethanol. So I'm going to pause for a second. Okay, so this is yeast. Okay, so yeah, so we have anaerobic. This is ethanol. It's done by yeast. It produces CO2, ethanol. And then animals, they produce lactic acid. And when you're working out and you're sore, this is what you feel. This is the burn. When you feel that burn from your working out, you're building up lactic acid. That's anaerobic respiration. That's really it. That's really all you need to know. Okay? The yeast, using yeast, we use this in obviously in baking. We use it, baking. And then we also use it to make ethanol. It's kind of all there is to it, guys. Oh, sorry. The last thing I know I was forgetting, the respirometer. So this is this right here is the concept, right? You have anaerobic, can be in yeast or animals, and it's in yeast it produces ethanol and CO2, and animals produce lactic acid. Aerobic is obviously, we covered that, right? So the last thing is a respirometer. What is a respirometer? So a respirometer, it has a few things, okay? Basically, what is the formula, right? We have C6H12O6 plus 6O2 yields 6 CO2 and 6 H2O. So we can measure two things. We can either measure the usage of oxygen, or we measure the production of CO2. Okay? Now, we need a few things. Basically, we need some kind of hydroxide. So normally, we would use potassium hydroxide. Okay? Now, what this does, this solution is real simple. It absorbs CO2. Okay? And you need something doing respiration, so we need an organism doing respiration. Now, this can be an animal or a plant, right? So we got to have some kind of chamber. So generally, you have like 2 different chambers, right? And they're connected by a capillary tube between these 2 chambers. So in this location right here, we have some kind of organism. I mean, this can be peas, it can be seeds, it can be you can put mice in there. You could put like grasshoppers, insects. So you can use anything in here that's respiring, right? Now, what's going to happen is that below this, we have our potassium hydroxide, our KOH. And this is going to absorb CO2, which means, what are we measuring? We're basically measuring the usage of oxygen. So this tube right here is filled with water. And in this chamber here, oxygen is being used. So oxygen is going down, and the carbon dioxide is being absorbed by the potassium hydroxide. So this water level should move this way because the oxygen is being used inside the chamber. So there's less oxygen. The O2 is being absorbed by this, and so the water will move up. The water between these 2 chambers will slowly move up, right? As the air inside this chamber is being used, the water level moves up, and we can measure the rate of respiration. That is a respirometer. So we're measuring the consumption of oxygen. There's plenty of pictures of this online. There really is. Please Google them. I don't really, let me try to draw, real basic respirometer. I'll draw one real fast. I just real fast Googled it, right? So you just got to have, actually, it's not much different than I just drew. So, let me pause. Okay, hold on, what happened here? Hold on, pause again. Okay, So I just Googled an image real fast. So this is the first image I came up with. This comes from Bio Ninja. So they do it the same way. You have two chambers. You have your two chambers. You have an equal volume on the right side. As the left side, you have your absorbent. You have some kind of hydroxide to absorb CO2, and you have your specimen in the middle. So as the oxygen is consumed here, the water level is going to change, and you can measure respiration. Now, because paper 3 is canceled, they're not going to ask you, like, they could ask you how to design an experiment. They could ask you, like, what would you use? Like, what would be your variables? So, like, look, you could apply fire to this. You could have fire here. Yay, fire. Where's fire? Red for fire, right? You could do temperature, right? You could do temperature. You could have 5 different chambers and different species, and you could be like, okay, is a cricket or a grasshopper or a beetle respire faster? You could make a mice run on a treadmill and see if they'd go fast. You know what I'm saying? There's so many you can do. You gotta do something to the specimen, right? This is just a setup. So you could do temperature. You could do. You could take germinations. You could take seeds and zap them with radiation like a microwave and be like, okay, you have 5 different chambers. You have 5 different seeds. They've all been exposed to different levels of radiation, and you want to see which one's gonna have a faster rate of respiration. So that's SL respiration. You gotta know a respirometer, and you gotta know anaerobic. You gotta know what ATP is. You gotta know the cycle. You gotta know the word. You gotta know the word equation for respiration. They like to ask about this. You know, like what's the purpose of the alkali solution? Is an alkali solution? Okay, this is 2.8 in a nutshell, guys. That's really all there is to it. Yeah, so don't worry about this plant. I don't know, just in anaerobic respiration, just to recap, all you gotta know is 2 things. What happens in animals? Animals produce lactic acid without the presence of oxygen. When you're sprinting or weightlifting and you run out of oxygen, animals resort to lactic acid lactic acid fermentation, and this makes ATP, but it's short term. This is a short term. It's a stopgap. Okay, and then anaerobic, we use yeast, and yeast take glucose and make CO2 ethanol. So we use this obviously in baking and other things as well. And that's really all there is to it. All right, guys.